This lesson is about lipids, and here are your objectives. Now, in contrast to polysaccharides, polypeptides, and proteins, and nucleic acids, lipids are not actually polymers. They're just sort of a grab bag of molecules that have in common that they are all hydrophobic. So lipids are a group of hydrophobic molecules. First, let's look at triglycerides. Triglycerides are also called triacyl glycerols because they are made up of one glycerol and three acyl groups. And we commonly know them as fats if they're solid at room temperature, or oils if they're liquid at room temperature. And they all have this kind of structure. They have a glycerol, which is a 3-carbon alcohol molecule joined to 3 fatty acids. And they are hydrophobic overall. And here we see a molecular model of a triglyceride with glycerol and 3 fatty acids. Now, triglycerides are grouped based on the composition or the type of fatty acids they have, either saturated or unsaturated. In a saturated fatty acid, all of the carbons have as many hydrogens on them as they could possibly. So if I draw a structure like this, where every corner represents a carbon and the hydrogens are just implied, every single carbon is full of hydrogens and there are no double bonds. In contrast, an unsaturated fatty acid has double bonds, and those double bonds mean that it can't fit as many carbons, and that's why it's unsaturated. So let's say there was a double bond there. Unsaturated, again, has double bonds. Saturated does not. Now, you can have monounsaturated fatty acids with only one double bond, or polyunsaturated fatty acids with multiple. So they can be monounsaturated with only one double bond, or polyunsaturated with multiple. Here we see a polyunsaturated fatty acid with multiple carbon-carbon double bonds. Notice that the last carbon on the farthest end from the carboxyl group is labeled the omega carbon, and 3 carbons in from that we see a double bond. That makes this an omega-3 fatty acid. Humans can't synthesize omega-3 fatty acids, so they have to take them in their diet. Now, saturated fatty acids, because of their relatively straight structure, can pack quite closely together, and as a result, they're quite dense, and saturated fatty acids, or triglycerides with saturated fatty acids, tend to be solid at room temperature. So think of animal fats, which are high in saturated fats, they're solid at room temperature. In contrast, unsaturated fatty acids, which have this carbon double bond, tend to have kinks in them, as we'll see in a moment, and that prevents them from packing closely. Therefore, they're less dense and they tend to be liquid or oils at room temperature. So unsaturated fatty acids in triglycerides give us oils, whereas saturated give us fats that are solid at room temperature. We can further classify unsaturated fatty acids and the triglycerides made out of them into cis and trans fatty acids and triglycerides. In a cis fatty acid, the double bond has the carbon backbone extending onto the same side of the double bond on either side of it. So here's the carbon-carbon double bond, and we see that the carbon backbone extends on the same side on either side of it. And this prevents close packing, like I talked about before, and decreases density. The vast majority of naturally occurring unsaturated fatty acids, unsaturated triglycerides, are of the cis variety. But when we hydrogenate unsaturated fats, meaning we take unsaturated fats that occur naturally and we force hydrogens onto them to try and make saturated fats for example, baking products, then we can end up with trans isomers. So these are artificially created, and the carbon-carbon double bond has the carbon backbone extending on different sides from it, and it doesn't lead to the same kind of kink here as we saw in the cis fatty acids. Now, trans fatty acids, trans fats, have been associated with an elevated risk of cardiovascular disease, and so they're banned or significantly limited in food products now. Here's a model of another fatty acid. Here you can see that all the double bonds are cis, which is producing kinks in this fatty acid. Also notice that if you count from the omega carbon, or the last carbon, 6 carbons in, you get to a double bond. That makes this an omega-6 unsaturated fatty acid. Now, if you take a triglyceride, remove one of the fatty acids, and put in place a phosphate, you get a phospholipid. As we've seen before, phospholipids are key in the cell membrane. They make up the vast majority of the cell membrane, and they form a bilayer because they have a hydrophobic end and a hydrophilic end. So the hydrocarbon tails of the fatty acids make up the hydrophobic end because they're nonpolar, and the phosphate end, which is polar, makes up the hydrophilic end. So as we've seen previously, overall the molecule is amphipathic, meaning it has a hydrophilic end and a hydrophobic end, but we still classify it as a lipid. Lastly are steroids, and steroids all contain a carbon skeleton of 4 fused rings like this. Examples include cholesterol, which as we've seen is an important fluidity buffer in animal cell membranes, and then from cholesterol other steroids like steroid hormones are made. Steroid hormones include male sex hormones or androgens like testosterone, female sex hormones or estrogens like estradiol, and then others as well like cortisol, which is a stress hormone, and aldosterone, which is involved in regulating salt balance. So that's an introduction to lipids.